Hello, I'm Dr. Malescu, and today's lesson number one, is the foundational aspects of microbiology, teaching you about the history, understanding some basic concepts before we begin our journey in our 5-week microbiology course geared towards people in the medical world, the surgical technology program. So let's take a peek. The first lesson is literally from your book. The PowerPoint, yeah, it's a little bland, so I'm going to make it a little bit more exciting. So all I want to say is this: the background of microbiology begins with the fact that we can't see these organisms, right? So over a century ago, we figured it out with the latest and greatest in technology. Of course, this started way many centuries in the making in terms of discovering pathogens and organisms that are found in our body. So the three different types of microscopes that we currently use are the basic compound microscope, the scanning electron microscope, and the confocal laser microscope. The one most commonly used in large colleges and universities, as well as clinical settings such as private offices and, of course, hospitals, is the basic compound microscope. You could read a little bit more detail about that in the book. For the sake of simplicity and outlining, and of course being able to get assessments out of these chapters that we're reading, I'm giving you the highlights. That's all we're doing. This is micro learning, giving you the highlights. What should you know? You should know the details, okay? You should definitely know the details of these three types of microscopes and what they do. Now, another detail that you might want to know is that the microorganisms, when they don't create disease and are normally found in a certain amount that is not pathogenic, okay, causing disease, that's called normal flora. So bacteria are everywhere. We have bacteria that's good bacteria that act symbiotically with our bodies. So microorganisms that are beneficial, like lactobacillus. We have E. coli, that if it comes out of balance and there's overgrowth in the culture of E. coli, then you get an infection. An infection is overgrowth of a bacteria that is out of control and needs to be destroyed by an antibiotic. All right, so what is normal flora? Again, I'm revisiting that, circling back to normal flora. Normal flora can act symbiotically with an organism to create balance. So we have normal flora in the skin, such as Staphylococcus aureus, which act as a symbiotic relationship with us. If it gets out of hand and there's overgrowth, then you have a skin infection. Same thing with E. coli, same thing with lactobacillus. And when we encourage people to take, for example, yogurt and eat yogurt while taking an antibiotic, it's because we are losing that nice balance where you have a healthy culture of bacteria in the gastrointestinal tract that is involved in your digestion. And when you're taking antibiotics, perhaps to kill some other organism that is causing an infection, like E. coli, if there's overgrowth, it's also going to kill the good bacteria like lactobacillus. And that's why we eat yogurt with natural cultures of bacteria that keep us healthy, or we take it in capsule form that we keep refrigerated so that that flora is something that we can repopulate in taking that capsule. We can repopulate in the gastrointestinal tract for our benefit. All right, now the bacteria that cause disease are called pathogenic bacteria. Of course, we are focusing right now on bacteria, but throughout our journey in microbiology, we will be talking about bacteria, we will be talking about viruses, we will be talking about prions, which are protein structures on their own in small amounts, just like normal flora, are not virulent. Okay, they don't produce an infection. However, in large amounts, they do. So there's prions. And then finally, last but not least, are the parasitic infections, the worms, the tapeworm, and various other parasitic infections like, contaminated water can have parasites, and that is very common when you leave the United States if the water is not treated. In addition to that, we have the fungal world. The fungal world can be very beneficial to us in terms of medicinal purposes, but also fungus among us is not pleasant. I like to always joke around about that because I was a podiatrist, and so I saw fungus among us a lot. So fungal nail infections are very hard to treat, and we have oral antifungals. We also have topical antifungals for fungal skin infection like tinea pedis of the skin and the feet, tinea cruris, which is infection in the crotch area. All of this is due to moist, dark places. That's what fungus likes, if it's moist and it's dark. So obviously the groin, the underarm is a common place to get fungal infections. And of course the feet when they sweat a lot and they're in a warm, dark, moist place like socks and shoes. And of course, if you get your feet wet and you keep them wet in the shoe, such as soldiers wearing boots, they get trench foot. These are all different types of infections that can take place. All right, so I've said too much. This is just supposed to be a micro learning session in which I'm just describing chapter 1 and 2 for you so that you are able to read the chapter, do the lessons online, and be able to take your first quizzes and all that. Okay, so the next thing I want to talk about is classifying microorganisms. So the bacterial world is divided into actually, I should say the animal world, because really the organisms can be one-celled or many-celled. So, all right, so let's go into that. We have the protists, which are the one-celled organisms, which can be divided. So we have two groups, the prokaryotes, which is the bacterial world, and the mycoplasmas. And then the eukaryotic world, which is the animal world and the plant world, that is very, very diverse. And we're not going to go over that, obviously, in this session. Just make sure that you understand the differences between the prokaryotes and the eukaryotes. For the purposes of microbiology, you'll be talking mainly about prokaryotes and how bacteria affect our bodies as eukaryotic multicellular organisms, because we as humans are part of the animal world. We're mammals, and we are obviously multicellular with multiple organs and organ systems making up our organism, the human. Okay, in talking about classifying viruses. So viruses are a little bit different than bacteria. They are not capable of reproducing on their own. They can't reproduce on their own, unlike bacteria. They're not independent. They are very dependent on us. And that is why they're dependent on infecting us and replicating inside of us. That's the only way that they can live. So remember that they are parasitic in that sense. In other words, that they need a host cell to be able to replicate. Okay, there are so many viruses out there. There are specialists, virologists, that just simply study viruses all their life. You can get a doctorate just in virology, okay? But microbiology is a more comprehensive umbrella study of all the different types of microorganisms. Virology is just simply studying the viruses, okay? And then immunology is simply studying the immune system and how it can defend or fall prey to bacteria, viruses, parasites, etc., fungus, okay? All right, so the viruses we have are measles, HIV, common cold. Of course, we have way more than that. Just giving you some examples here in this PowerPoint presentation. Of course, please understand that this is by no means you watching this video is going to make you pass your first assessments of week 1. You really need to do the legwork, read the book, take notes, watch this PowerPoint presentation, go through the exercises in the online book, and then you will be able to master these first 2 chapters. Okay. Let us dive deep into prions. I don't know if you know about the people in the past, probably over a century ago, it's not done anymore, but there was a tribe in Australia, indigenous tribe, that it was part of their culture, and you need to respect and understand that this is what was done because they didn't know any better. They didn't know about microorganisms. In order to honor their family members that died, they felt that by eating their brain, they would also take in the soul of their family members and remember them. It's very unique and strange to us, but that was their belief system. So they would eat human brain. Okay, so why am I mentioning that here? Well, because prions were present in these brains. Now, you could also get mad cow disease, which is basically prions as well. These are infectious proteins that by themselves, they don't do much, but if they're in a large amount and they're ingested in a large amount, such as meat, then you will take on those prions that get out of control and, of course, cause infections of the central nervous system, really, really bad to get. Brain damage, and yes, death can take place. Okay, sorry to be a Debbie Downer, but don't eat infected meat. Prions. Prions is what you worry about. Okay, so unfortunately, we can't make them integrate them into the category of viruses or bacteria. So we made a new separate category just for prions. Okay, they don't fit any cellular category of description in microbiology. They are anucleate, they don't have a nucleus, they cause CNS infections, and they're really, really basic. They don't even have nucleic acid substructure that can turn into an RNA or a DNA, such as in viruses. And of course, bacteria, all right, which have DNA. And of course, us, we have DNA, all right? We already know all about that. So I hope that you understand that this is a whole separate category that can cause infections. Not as common as bacteria and viruses, but nonetheless, they exist, and we do need to know about them in microbiology. All right, next, we're going to talk about the structure of the eukaryotic cells. So the eukaryotic cell has a cell membrane with an outer layer that is selectively permeable. In other words, certain things come in and things come out. Only certain things come in based on these proteins on the membrane. We call them receptors, okay? And they have channels that only open up when there's molecular binding, like a lock and key method, or like a hormone can attach to a receptor, like insulin attaches to the insulin receptor, then the channel opens and glucose enters into the cell, things like that. So that's what selective permeability is in the cell membrane. So yes, we do need to know that throughout your medical world. In your medical profession, you're going to learn a lot about cell structure and function. I know it seems boring now, but the way we study new medications out there and how they properly enter the cellular membrane, super important for being able to combat these microorganisms such as bacteria, viruses, parasites, fungi, and of course, we recently learned about prions. Okay, now what about the nucleus? The nucleus is the motherboard. So for example, In a computer, the motherboard, if you take out that, you have a box that's rendered useless. So the motherboard of the cell is the nucleus. It is the control center. It contains the genetic information so that the organism can replicate itself, whether it's a DNA or an RNA. So for example, viruses are very simplistic. They don't have DNA, some of them. So they, you have RNA viruses that will need, like HIV, will need an enzyme called reverse transcriptase to change up the molecules into a DNA structure that will integrate into the DNA of the human being. And so that diabolical HIV virus integrates itself into the DNA of the human, so that every time the human cell replicates, so does the HIV cell. So does the HIV virus, the components of the virus. Okay, and that's what HIV infected cells do. They take over, and it basically, HIV infects the T cells, which controls your immune system. So that's an example right there as to why, if you get a hold of the motherboard, you pretty much control the world. It's like putting a virus in software, or downloading malware, and now your computer is a box that's rendered useless because it has a virus. Same concept. All right, now let's talk about eukaryotes a little bit more besides the nucleus and the cell membrane. What I want to mention, let me just move my screen over so you can see the spelling here. Eukaryote, this is how you spell it, E-U-K-A-R-Y-O-T-E-S. So eukaryotes, the eukaryotes, in addition to having a cell membrane and a nucleus, we have the semi-fluid, kind of like gel, right? So if you ever had gelatin, fruit jelly, not many people like it, but anyway, if you do like it, you know that you have the fruit that is inside the jelly that's kind of shaky. The jelly is shaky, it's kind of liquidy, but it's kind of semi-solid. That's what our cytoplasm is. It has fluid, but it's semi-solid, it's semi-fluid, somewhere in between, maintains your shape of the cell, and it is a space that not only contains fluid, it has cytoskeletal elements to maintain the shape, which are made of protein linkages. And of course, it has all the necessary components that are important in the function of a cell. So those different types of components are having a function that are independent of all the other components. Okay, so for example, we talked about the nucleus. There's also the mitochondria, which is the powerhouse of the cell, making adenosine triphosphate, making energy for your cells. So these are all the different components. Okay, so the nucleolus inside the motherboard, the nucleus, is a protein manufacturing company. That's all. We're making ribosomes in the nucleolus. All right. The ribosomes are helpers. They assist in protein synthesis, making a polypeptide chain of amino acids, which become folded into proteins. Proteins could be anything and everything from the genetic structure that defines your eye color, to enzymes that are needed in biochemical reactions in your body, like the enzyme lactase, which breaks down lactose, which comes from your milk. And that's why as we age, we start losing some of these manufacturing components in the nucleus. Your DNA starts to lose some of the genes, which are nucleotide bases of 3 that define the genetics, the genetic code to define a set of enzymes per se, or a particular phenotypic or genotypic characteristic in the human being. All right, so I've elaborated a lot, but basically all you need to know for the purposes of microbiology is that ribosomes assist in protein synthesis. They are made in the nucleolus, okay? And the nucleolus is a circle within a circle. In other words, let me just draw this out. if you have the cell, inside is the nucleus, and then within the nucleus is the nucleolus. So I'm filling this in. That's the nucleolus filled in nice pink. Okay. So that would be the nucleolus. Okay. And then the nucleus is where you've got your genetic information. And the nucleus has these little holes called nuclear pores, and your DNA, so I'm going to pretend these are little chromatin, which is the DNA wrapped up in histone proteins that, when unraveled, become chromosomes. And those are located in the nucleus, the cytoplasm of the nucleus. All right, we call it the nucleoplasm, which is basically the cytoplasm of the nucleus. All right, and then all the other organelles, such as the mitochondria, which provides energy, ATP, which I mentioned. All right, this is your cytoplasm, and your cell membrane, which is selectively permeable. And then your ribosomes are located on this serpentine looking structure that I like to say that it looks like pancakes. I'm going to draw it in a different color, the ribosomes. So this is called rough endoplasmic reticulum, and you got the ribosomes attached to this rough endoplasmic reticulum. And this is where we have protein synthesis, and the ribosomes are assisting in making proteins. Okay, I'm not going to go into huge detail in this lesson because I have on my YouTube channel an entire YouTube just on understanding the process of protein synthesis. Okay. All right. So that's that. I hope that that little diagram helped you in terms of visualizing what I'm talking about. So I am now going to erase this, and I'm going to move on to the next slide. All right, so the definitions, as I mentioned before, be sure to remember that your mitochondria is the powerhouse of the cell. Very cliche term, but it works for me. It works for everyone. You learned it probably in grade school. 7th and 8th grade is where you first get introduced to a little bit of biology, and you hear this terminology, powerhouse of the cell. Well, it is. It really is the battery of the cell. Remember it that way. ATP is called adenosine triphosphate, and it is a excuse me, a very large molecule that assists in creating the energy needed, the fuel needed for the cell. So this is how you spell it: adenosine tri, because it has 3 phosphates. Excuse me. Okay. All right. So that is adenosine triphosphate. Now, what I didn't mention in the previous diagram that I drew out is the Golgi apparatus. I like to call the Golgi apparatus the post office. Why? I'm going to write this down. Why? I'm going to draw out again the diagram, and I will tell you why I like to call it post office. When you look at my protein synthesis video, YouTube video, which I'll post for you in the announcements alongside this presentation, you will note that, as I mentioned, that your DNA is here. Your DNA gets replicated by the RNA, and it goes to the rough endoplasmic reticulum with your ribosomes. Okay, I'm not going to tell you the whole detail because that's for that other video, protein synthesis. Nonetheless, we make protein, okay? We make this chain of polypeptide chain of amino acids that leaves the rough endoplasmic reticulum, which I'm going to write here, rough ER. So lowercase r, uppercase E, uppercase R. So the rough endoplasmic reticulum is this pancake structure. The purple are the ribosomes, and we're making, this is the area of protein synthesis. Okay, we're making a polypeptide chain of amino acids. All right, well, what happens after that? I'll tell you. The polypeptide chain of amino acids it needs to be exported. But first, guess what? It goes to Golgi apparatus. So I'm going to draw another set of pancakes, but it looks very different. It looks more flat. And this is the Golgi apparatus. So I'm going to call it GA so I don't have to spell it all out for you because it's in your notes over here, Golgi apparatus. So that's your Golgi apparatus. In yellow are the polypeptide chain of amino acids. Well, you have a chain of amino acids, amino acid, alanine, lysine, valine, okay? And so, but it needs to be folded up quaternary into this nice little package. Well, where does that happen? In the Golgi apparatus. What happens in the Golgi apparatus? Packaging and delivery of proteins. So first the packaging occurs, thus the post office, right? Packaging. And once it's packaged, these Golgi vesicles come out of, they separate like via exocytosis. They will fuse with the cell membrane and leave with the protein to be exported to wherever it needs to go. Okay, that's the short of it, but watch my video, and if you want the details, you can get the details, but that's the highlights of protein synthesis. All right, so let me erase this, and I will move on to prokaryotes. So prokaryotes are just like we mentioned with eukaryotes, they do have a cytoplasm, but we call it more like a cell wall. The difference between prokaryotes and eukaryotes is this big difference, and you need to remember this right here. No nucleus. Okay, so a bacteria is a blob. It could be oval shape or round shape, and then you just got your DNA. Okay, or RNA, but it's mainly DNA. Okay, RNA is more, think about more like viruses. All right, so we're just going to say DNA. We have a very simple structure called nucleoid, which will store the DNA, but not as complex as a nucleus. And some of the bacteria will have a slime layer, which increases the resistance to antibacterial agents like antibiotics. All right, so I hope that was clear. Prokaryotes will carry out little tiny cilia, which are fine, tiny little filaments that move the fluid across the surface. Flagella are longer filaments that will increase the motility of the cell. Obviously, eukaryotic cells that have this are the sperm cells that are involved in reproduction in mammals. And then, of course, prokaryotes can also develop spores, which is almost like they go into a shell, like a peanut shell, a hard outer shell. And it inactivates the bacteria, and they become active when they embed themselves into an organ or an organ system. And then at some point they break free and they activate and they cause harm and havoc. And that's the tuberculosis bacteria. In its active form is highly contagious, airborne. But you can also inhale the spores. The spores could lay in your lungs for years and one day just activate. And that's why we learn about spores in microbiology. And we need to learn about spores so we know how to treat them. We have different antibiotics that can obviously combat this type of bacteria that builds up spores. Okay, now, normal flora, as I mentioned at the beginning of this lesson, is not going to cause any infection unless it grows out of control. So it exists on us, skin, in us, or around us, and may become pathogenic if they do not remain in its intended location on the body or in the body, and of course if they grow out of control. So typical example, E. coli should not be in the urinary tract. All right, it is normally found in the gastrointestinal tract. Children that do not know how to wipe properly can inadvertently cause themselves havoc and infection by wiping their poop with the toilet paper and then introducing it to the urethra, whether they're male or female. And it's obviously more common in females because of the proximity. Anyway, I got a little detailed on that, but you can imagine what can happen. So there's various ways that people get infections from normal flora, okay? So these are some of the things that we can think about in microbiology, learn about, and figure out how to treat it. So UTI is very treatable with antibiotics. When we dive deeper into antibiotic therapy, you're going to learn about the different antibiotics that treat urinary tract infections, because you will have to know it. All right, so make sure you understand the term. It's in your discussion board: virulence, pathogenicity, and normal flora. So the next term that you need to know is pathogen. The definition, I mentioned it throughout this lesson today, right? A pathogen causes disease, okay? Even normal flora can be a pathogen. If it's not located in the right location, it shouldn't be there. It can cause infection, or like I said, if it goes out of control, you know, E. coli gone wild per se, or Staph aureus gone wild, you have a Staphylococcus aureus infection. So what are the ways that we can fight off these pathogens? Well, the first line of defense is your skin, protective barriers like the mucous membrane in your nose, your skin, as I mentioned, your pH. So like in women, the vaginal pH, it's acidic. It is for a reason that the pH of the vagina has to be a certain pH to avoid overgrowth of yeast and fungus and introduction of parasites and bacteria. I don't mean to be disgusting, but that's what happens. So unfortunately, the more openings you have, the more vulnerable you are. So that's why we are vulnerable through our nose. This is how we get the common rhinovirus infection. It's the common cold that gets in through our nose. We inhale in the virus, okay? This is why we wore a mask during this pandemic. Whether it works or not doesn't matter. It definitely reduces the infectious rate. And it, of course, if you have a better fitting mask, like a N95, then we're good to go. Obviously, you're going to learn all about protection because you are going to be in the OR. All right, now let's go over other ways of defense. We went over the protective barriers like skin, mucous membrane, and all our openings, you know, making sure that our pH is at a certain level. Even the pH of the skin has a certain acidity. Keratin is protective of the skin, so etc. You can read more about that as you read the chapter. Let's move on to your circulation. So your circulation is your blood. Your blood transports white blood cells. The white blood cells, there are different types of white blood cells. There's a mnemonic called "Never let monkeys eat bananas." Never stands for neutrophils. These are elevated when you have an acute infection. They are the first responders, the EMS of all white blood cells. They're called neutrophils. Those are never. Let, lymphocytes. Lymphocytes, you have the B cells, which release the antibodies, the T cells, which are direct hit. These are the foot soldiers that fight off infections such as viruses. Okay, so never let. Lymphocytes. B lymphocytes release antibodies. T lymphocytes are a direct fight against pathogens. Okay, so never let. Monkeys, monocytes for chronic infections. You'll see them elevated on a white blood cell CBC with differential. It's a complete blood count. That's what we call it. So if you ever, you know, you're going to work in a hospital, you're going to see a lot of the workups include CBC with differential. All right. So we went over never let. Monkeys, monocytes, chronic infection, monocytes, because they turn into macrophages, which, like Pac-Man, dating myself, will digest pathogens. Okay, so never let monkeys eat. Eosinophils. All right, these white blood cells are going to fight off parasites. Okay, so they'll be elevated when you have a parasitic infection or when you have an allergy. So eosinophils are elevated chronically, then you probably have chronic allergies. That asthma, or if none of those are relevant to the patient history, got to ask them about where they've been. If they've been in a lake, they might just have a parasite. Okay, so never let monkeys eat. So we just went over eosinophils. Bananas, basophils. So basophils are the least common, 1 to 2% of the population of white blood cells, and these release histamine and heparin, and so they're big in blue cells, and they are there as an allergic response. So there, I said it. Those are all the different types of white blood cells, and so these white blood cells circulate in your bloodstream, and thanks to maintaining your blood pressure, the blood cells are moving around to all parts of your body, to all the organs and organ systems. All right, so that's the second portion of the 3 lines of defense. And then the innate and adaptive response. So the innate is what you're born with, and the adaptive is basically your immune system kicking in when you get an infection. All right, so the protective barriers, like I mentioned earlier, intact skin and its secretions, the respiratory tract, for example, mucus. Your mucus is going to catch the virus or the bacteria. This is why it's important to blow your nose when you're sick and not to swallow it. Okay, get rid of the mucus. Expectorate, because chances are that mucus is your protection. You may not like it because your nose is runny and you're sneezing, but you're actively getting rid of whatever you have, if it's a virus or bacterial infection. The gastrointestinal tract, coming from the mouth to the anus, will have protective ways. For example, your saliva is going to have antiviral and antibacterial components, and immediately will release antibodies as well. Acidic pH of the stomach is going to kill any kind of pathogen. Example in the gastrointestinal tract would be different, they're called Peyer's patches, different patches across the intestinal tract with patches of lymphoid tissue that contain white blood cells to fight off infection. All right, then there's the genital urinary tract. By expulsion of the urine, the propulsion of urine is going to also release any kind of pathogen. All right, the phagocytic cells are the neutrophils. As I mentioned the neutrophils briefly in the Never Let Monkeys Eat Bananas, neutrophils are the first and foremost the EMS. If you cut yourself, or if you introduce a, let's say you step on a nail, okay, and it has bacteria on it, the neutrophils are first to respond, and they come in and, you know, besides the clotting mechanism occurring to stop the bleeding, the neutrophils will be present. Then if the infection is there for more than 48 to 72 hours, you're going to have the monocytes, I would say, transform or differentiate. I like the word transform, right? Because I think of the movie Transformers, right? They transform, but really in the microbiological world, we say differentiate into the larger cells called macrophages. Dendritic cells have these extensions, which are really cool, that you can see on a microscope, you know, with a prepared slide. You can see the dendritic cells literally picking up with their pods the pathogens. Lysosomes. What are lysosomes? These are found throughout the cells. They are very common in bone marrow and liver, and they roam around, and they have this release of enzymatic activity to get rid of foreign substances, viruses, bacteria, etc. Your innate immune response exists when you are born, okay? It exists at birth, but here's the catch. Unfortunately, it is a very general, should I say, a general protection. It is not specific. So non-specific protection, general pathogenic specificity, general pathogen involvement, but it's not specific. It's not pathogen specific. That is a problem. Okay, so this is how children will develop their immune system by more exposure. They develop the antibodies against each and every single pathogen that they come across. So it's not necessarily a bad thing. Will they get sick a lot? Yes, but that's how you build the immune system. You can't build an immune system by keeping someone in a bubble. So first exposure, it takes them 7 to 10 days, depending on the virus, to develop the antibodies. The antibodies, usually by day 3 or 4, you're starting to find out exactly the structure of that virus, and then your body will produce the appropriate antibody from the B cell that has been activated by this particular pathogen. So anyway, the innate immune response will kill invading microbes, but they're non-specific. It begins with edema, which is swelling, erythema, which is redness, okay, and pain in Latin means dolor, and these are the medical terminology words that I would like for you to get used to. Don't just say redness, say erythema, swelling, edema, and pain is dolor. So let me write those down for you. So redness is erythema. Swelling is edema. Pain is dolor. Okay? All right. So adaptive immune response, it takes 48 to 72 hours. Of course, it depends on if it's first exposure. If it's second or third exposure, you might already have an adaptive immune response within 24 hours, and the person may have minimal symptoms or completely be asymptomatic. And that's what's happening now with the spread of COVID, because so many people have been exposed to COVID during this pandemic, whether it was through the vaccination and boosters or whether they actually had it naturally. They are now somewhat immune, and some of them, some of the people may only have COVID-like symptoms for 24 to 48 hours. Some people may not have any and be inadvertently spreading it because, you know, typhoid Mary, that's what happens. It happens in not just COVID-19, it happens with all other kinds of pathogens. Look it up, typhoid Mary, that's how we first discovered that you can be a super spreader and be completely asymptomatic, but spread it to other people that are not immune. All right. Let's go over adaptive immune response. So the adaptive immune response is antigen presenting cells, APCs. So antigen presenting cells are the cells that are going to present to other cells. Hey, by the way, knock, knock, we've got a bad intruder here into our body. So it's presenting the intruding microorganisms to the T cells, which I said is basically the foot soldiers that are going to go in and kill the organism, whatever it is, whatever pathogen it is, okay? An antigen is a pathogen, antigen, pathogen, that's how you remember. So the antigen is the offender, it's a foreign body, whatever it is, whether it's a virus, bacteria, parasite, whatever it is, okay? It's marked as foreign, the immune system marks it as foreign, and the phagocytic cells will start to engulf the pathogen, try to swallow it up, and then present its fragments, all right, to the adaptive immune system, including the T cells, and say, hey, T cells, this is what we just killed. Can you go out and find some more of these? Because we need this person alive, okay? That's your immune system in a nutshell. So the adaptive immune system is cellular mediated. Without the APCs, nothing happens. So your antigen presenting cells are going to present to the T cells, and then the T cells get activated. It's like, hey, sounding the alarm. Okay, you've got a war going on. There's the war trumpet, and the soldiers are lining up, and they're like, okay, we have identified the enemy. The enemy was presented to us by the B cells. All right. And now we must march on, go into the bloodstream and start killing these mofos. All right, so that's how the immune system, I know it could be very boring reading about this, but that's how it works, okay? We can make it fun. Now, the humoral response is quite different. This is not like direct fight, direct killing, shooting, right? We are talking about the B cells saying, hey, you know, we got a better way. Okay, the soldiers can do their thing and go head on with these pathogens, but we have a weapon of mass destruction here. And so the weapon are the antibodies, okay? So the T cells will interact with the B cells that produce the antibodies. The antibodies are called, another name, immunoglobulins, all right? So antibodies, otherwise known as immunoglobulins, is basically your humoral response, okay? Meaning we have a weapon, we're going to use it against the pathogen and destroy that pathogen, which we defined already in the immune system as the antigen. An antigen is a molecular structure that is foreign, and the antibody or the immunoglobulin is going to bind with it and destroy it. And that is like I said, a weapon. Whereas the T cell is direct head-on fight with the pathogen. So I do hope the analogy helps. So an infection is when a microbe has caused an invasion, and the invasion can be localized by creating that erythema, edema, all right, dolor, because when you have pain, you're not going to move. If you move, you're going to increase your circulation, and the risk of introducing the bacteria or other pathogens into the bloodstream goes up. So there's that. Okay, so you can have a local infection or an extensive infection along a fascial plane, such as cellulitis. These are the things that you're going to learn in the medical world, so might as well learn it now. I like to teach microbiology from a clinical standpoint, and this is technically medical microbiology because we are teaching you all about these microbes and how they affect the body and how do we treat it, okay? So virulence is determined by the structure of the cell and the production of an endotoxin. So that's another topic. All right, so pathogens can produce endotoxins that are contained within that particular microbe, or they can release, all right, an exotoxin. So they're produced or secreted by the microbe. Either way, it is lethal and not good for the human or the animal exposed to this particular pathogen. So the pathogens that are more virulent, obviously, by these definitions that I just gave you, the more virulent the pathogen is, the higher the chances that it has an endotoxin or an exotoxin. Now, let me give you an example. We can talk about bacterial endocarditis. We have many viruses out there, including COVID-19 and all its other variants, that are very toxic. They have an endotoxin that can cause bacterial endocarditis. We can have streptococcal pyogenes, strep throat. If you don't control it with an antibiotic, it gets from the throat into the bloodstream, gets to the kidney, it can cause glomerulonephritis, which is inflammation of the little filtering systems in your kidney. And it can get into your bloodstream and cause bacterial endocarditis. Wow. So these pathogens can kill you or completely permanently damage your organ systems. All right. So get treated is my whole point. All right. So in the end, what have we talked about? The most common pathogens that we've talked about are bacteria, viruses, fungi, and parasites. Okay. So wrapping it up, we talked a little bit briefly about different types of bacteria. We talked about different types of viruses. We did not touch upon fungi and parasites yet, but we will dive deep. We'll do a deep dive very soon. This is just an introduction. It's your first lesson. So basically the last leg of this lesson number 1 is to talk about contact transmission. We can have direct transmission or direct contact with a person, So me speaking to you and perhaps some of my saliva coming out as I speak, or if you're sneezing or things like that, coughing is going to release lots of microorganisms, all right? So that is why we wore a mask during this whole pandemic. Now, what about indirect contact? For example, we can have a fomite, which is this wall, my computer. All right, so these are inanimate objects. All right, a vector is something that could be transferred from a fomite to a person, or a person to a person, like bird flu, a bird. All right, droppings, fomite, and then to a person. Okay, airborne droplets are another way. So these are all the indirect contact of getting transmission. Direct contact is sexual contact, is an example. Indirect contact is me, for example, sending out that airborne droplet. Okay, so these are the different ways of contact transmission. Direct contact through kissing or sexual contact, that is definitely a way to inoculate an organism. Indirect contact can be a fomite, and as I mentioned, vectors. So fomite is an inanimate object. So this is why it's super important if you're going to be a surgical technician or if you're working in the hospital. There's so many things to think about. In the forthcoming chapters, we'll discuss more detail on fomites, and that is why we sterilize instruments, okay? All right, moving on. When someone gets an infection, we talked already about the terminology, erythema, edema, all right, odolar, pain. But in addition to that, if it's another infection that is more severe, other than just a local infection, the whole system is going to turn on, the immune system. We're going to develop a fever, which of course, you know, it may not be pleasant for the person that's infected, but it does assist in clearing out the infection because lots of these pathogens do not like the high temperature. All right, so that is a defense mechanism of the body. Inflammation is another defense mechanism of the body. Of course, we mentioned that you know, the inflammatory response is great in killing pathogens, but it also unfortunately can do damage to organs and organ systems if the inflammation is supercharged. And that was the initial issue with COVID-19, right? And we talked about that. Dilation of the blood vessels. Why do we do that? Well, because we want to bring more blood flow to that space so that we introduce more white blood cells, the ones that we talked about, right? Never let monkeys eat bananas. The neutrophils, right? The monocytes, okay? Never let monkeys, the lymphocytes, all of those white blood cell types are moving to that location to fight off the infection. So infiltrating the infected site with antibodies. Who produces the antibodies? I already mentioned it to you, the B cells, right? The B cells release the antibodies, all right? So white blood cells will definitely combat the microbe. Unfortunately, lots of white blood cells result in death of the microbe, but some of them also die off as well. And there needs to be a cleanup crew to come in, and that's where the macrophages come in and digest all of the mess, the war going on inside your body. All right, so to summarize this lesson number 1, we talked about, let's circle back to what we talked about, and I want you to read the chapter. We talked about the category of prokaryotes versus eukaryotes. We talked about the different ways that we can look at microorganisms. Make sure you understand the 3 different types of microscopes. Make sure you understand the terminology pathogen, virulence, and normal flora. And that normal flora lives on or inside the body, and everything's fine as long as the flora is located in a specific location. If it moves to another location, such as I mentioned, E. coli is found in your gastrointestinal tract, has no business in the genitourinary tract, which can be introduced with improper wiping. So things like that, normal flora gone out of hand. Staph aureus, normal on the skin, but if it grows out of control, then you have a Staph aureus infection, okay? So that's pretty much it. The other second page of the summary is that you need to review in your book and understand before you take any assessments and your quiz, understand innate versus adaptive immune system. Humoral immune system is your antibodies being released by your B cells. Cellular immune system is your T cells, the soldiers going in on the field and directly attacking the pathogen. They only get activated when the APCs are telling them to get activated. What are the APCs? Those are the antigen presenting cells, all right, that have killed the pathogen. So it introduces the components of that antigen or pathogen, right? And so therefore the T cells get activated, all right? So pathogenicity is the ability of microbes to cause disease. Make sure you remember that we had endotoxins and exotoxins. We have bacteria that produce endotoxins within their cell and exotoxins, meaning the bacteria release toxins that are very damaging to the body, causing bacterial endocarditis, glomerulonephritis, things like that. And we also mentioned tuberculosis, which can form spores and lay latent in your lungs for years and then get activated to an active tuberculosis infection. All right, and then don't forget the term virulence is the strength of disease-causing microbe. That is on your first discussion board. I have already started to respond to some of you, so please get that discussion board out by tonight. All right, later on today or tomorrow, I will go over lesson 2. I do hope that you enjoyed this lesson. All right, see you next time. Bye.